This is my interpretation of research by P. Kurian from their paper Ultraviolet Superradiance from Meganetworks of Tryptophan in Biological Architectures. Introduction. Our everyday experience versus the microscopic realm. We often talk about the physical world, but within it exists a hidden layer, the quantum world. These terms might seem interchangeable, but they describe our universe on vastly different scales. The world we experience directly, chairs, trees, and even our bodies, is the domain of classical physics. This incredibly successful set of laws allows us to predict the motion and interaction of these objects with great accuracy. In this world, properties like position and speed can be precisely known, and cause and effect relationships reign supreme. The quantum world, on the other hand, deals with incredibly tiny, the building blocks, of matter like atoms and subatomic particles. Here the laws of classical physics crumble. Instead, the strange and wonderful principles of quantum mechanics and quantum biology take over. This realm introduces us to concepts like quantization. Imagine a staircase instead of a ramp. Certain properties like energy come in distinct steps, not smooth gradations. Wave-particle duality. Light can act like a stream of particles, photons, or a wave, blurring the lines between the two. Uncertainty principle. The more precisely you measure a particle's position, the less certain you become about its momentum, and vice versa. There's a fundamental limit to how much information we can know at once. Superposition. A quantum system can exist in multiple states. Think a coin being both heads and tails until a measurement forces it to choose one. These concepts might seem mind-bending, but experiments have confirmed their validity. They are essential for understanding the very foundation of our universe, including how we operate within it. The key difference. The physical world is the one we interact with every day, governed by the predictable rules of classical physics. The quantum world, invisible to our senses, dictates the behavior of the tiniest particles with its bizarre yet well-tested principles. Not separate realities, but connected layers. Importantly, these aren't entirely separate realities. The familiar world we experience is ultimately built upon the strange underpinnings of the quantum world. The seemingly predictable behavior of large objects emerges from the complex and probabilistic world of quantum mechanics and quantum biology. Biological processes possess both physical universe and quantum aspects. The efficiency of certain biological light-harvesting molecules or the function of specific enzymes involve both classic and quantum phenomena. I present to you an exploration into both worlds via the amino acid tryptophan. This is what I call quantum photobiomodulation via tryptophan superradiance. Now here's a better explanation of what that title means. Quantum photobiomodulation via tryptophan superradiance suggests a scientific concept or phenomena involving the use of quantum physics principles and photobiomodulation techniques to influence biological processes, mainly through the interaction of tryptophan molecules with ultraviolet light. Now, photobiomodulation involves the use of light to stimulate biological processes at the cellular level, such as promoting tissue repair, reducing inflammation, and modulating cellular metabolism. The amino acid tryptophan is crucial in protein synthesis and a precursor for various neurotransmitters such as serotonin and melatonin. Superradiance refers to a phenomena in which protons are emitted coherently, which means in a synchronized fashion, from a collection of excited molecules or atoms, leading to enhanced radiation compared to individual emissions. In the context of tryptophan, it suggests that when tryptophan molecules are exposed to certain light conditions, they may emit photons in a coordinated and amplified manner, potentially leading to significant biological effects. Think of a sporting event where somebody begins the wave and one person raises both arms up and then the next person does it and the next person, and then that flow keeps happening around the arena. This is exactly the same thing. So the tryptophan molecule will absorb light. One person begins to do it. That's the first tryptophan molecule. More and more people begin to do the wave. It's emitting more energy, passing it along and amplifying that energy from one person to the next to the next. So it's building momentum as well as amplification. That's what superradiance is. Now, overall, quantum photobiomodulation via tryptophan superradiance hints at a cutting-edge approach that combines quantum physics and photobiomodulation techniques to harness the unique properties of tryptophan and light for therapeutic or biological modulation purposes. The amino acid tryptophan contains an aromatic ring that is a chromophore. Now, if you look at the tryptophan molecule in the upper left-hand corner, you will see a ring with 3 lines. That is the aromatic ring, which is also a chromophore. A chromophore is the light absorbing or harvesting portion of specific molecules. Molecules with chromophores exhibit behavior analogous to an antenna interacting with radio waves. So instead of invisible radio waves being picked up by the antenna and then converting it into sound that we hear as music or talking, we get invisible light waves that are picked up by these chromophores and converting them to some sort of biological function. This gives tryptophan a remarkable capacity for efficient ultraviolet energy absorption. Tryptophan is found in transmembrane proteins, multi-tryptophan proteins like myoglobulin, which is a muscle protein, the oxygen-carrying portion of blood, hemoglobin, cytochrome C oxidase, which is the 4th cytochrome within the mitochondria, passing electrons along to number 5 guy, making ATP, which is energy. Cytochrome P54, which is a set of enzymes that are crucial for detoxification and cellular metabolism. Cytoskeletal filaments, which we'll talk about in a bit, such as centromeres, centrioles, cilia, flagella. Subneuronal elements, such as axons of nerves. Photoreceptor complexes like melanopsin within the eye and neuropsin and nerves. These are photoreceptor complexes that pick up on light and then transfer that light into energy and information that our body could use for light interpretation and nerve function. Viral caspids, the coatings of viruses and other cellular structures. Now, this slide, I won't get into the whole slide. You could review it at your leisure, but this is chromophore structure, function, and examples. A chromophore is a molecule fragment that absorbs specific wavelengths of visible or ultraviolet light, leading to the color we perceive in many cases. So anything that does exhibit a color does have a chromophore in it, and chlorophyll, for instance, green is a chromophore. Chromophores absorb light, and that translates into wavelengths, but then it always can emit light as well, and that is fluorescence. Now, the function, when a molecule containing a chromophore absorbs light, it results in excitation of electrons from a ground state to an excited state. It's almost like a potentiation of the effects. And at the bottom, you can see also, we talked about those aromatic rings, but the bond in between carbon and carbon, or this entire complex, absorbs light. Carbon to oxygen, the double bond, also absorbs light. So, and then as the aromatic ring is also called a benzene ring. So chromophores exist throughout our body as well, physically, as well as in compounds such as vitamin D, B12, melanin, every hormone, amino acids, tryptophan, phenylalanine, histidine, tyrosine, so thyroid hormones, as well as neurotransmitters, all are made up of chromophores. So it just, in a way, can show you the importance of light onto our body. In order for all of those things I just mentioned to work efficiently and effectively, we need to have light in our lives. All right, so first I want to draw your attention to the diagram in the upper right. You can see A, B, C, and D, and they're moving from a smaller architecture to a more sophisticated and denser architecture as it goes from A to D. Now, these four represent collective ultraviolet superradiance from very large protein architectures of quantum emitters, which are the tryptophan quantum emitters, and is enhanced with increasing size, resulting in observable and robust increases in thermal quantum yield. So as these architectures, think of it as linking more and more and more batteries and becoming more and more powerful, or adding a preamp to your stereo system to get more wattage to be able to crank the sound up higher. That's what this increased density of this tryptophan architecture can achieve. So to the text, from tubulin, which is A, the smallest tryptophan photoarchitecture network, we move to larger protein structures, tryptophan meganetworks, called microtubules. So number B is one microtubule, in which tens of thousands of these tryptophan molecules self-assemble to form even larger microtubule bundles found in centrioles, which is C, and neuronal axons, which is D. Centrioles, now numbered C, enable cellular reorganization and remodeling for mitoses, cell division, differentiation, transport, and a neat concept, allow for habitat exploration and apoptosis. Now here is a diagram of a centriole. And so if you look at the cell to the far left, the two centrioles form a complex called a centrosome. And if you look at the far right, you'll see a cell with the little yellow highlight inside of it, and that will be the centrosome made up of centrioles made up of microtubules. So again, light harvesting architecture. Now, here is also a centriole, but the centriole can change its shape a little bit and become something called a cilia. So the diagram on the left then has one little cilia sticking out of the cell, and it's a mechanical receptor. So it's picking up on vibration and acidity and electrolyte information. It's a sensor that sticks out of the cell. So that the cells in the center picture happen to be kidney cells. So it's picking up on all the stuff of the urine flowing through the kidney, so it could pass information onto each other cell as well as the rest of the body and communicate information. But the fact that it is made up of microtubules means that it actually can have an interaction with light at a quantum level. Now on the right, it's the same cilium, but it changes shape just a little bit more, and it becomes a photoreceptor like neuropsin and melanopsin in the eye or the nervous system. And it is picking up on light energy. Now this is the cilium dissected. Centrioles and basal bodies are structures comprised of microtubules found in most of our cells, which means if they're in most of our cells interact with light. Centrioles, together with pericentriolar material, form centrosomes. So remember, two centrioles make a centrosome. And basal bodies form flagella and cilia. Both centrioles and basal bodies have the same molecular organization and can be interchangeable in the cell. That is, a centriole can move near the plasma membrane and form cilia. So you can see on the left the diagram, it's the little antenna that will poke out of the cell that will pick up on various types of information depending on the cell that it is in. And a basal body can travel to the inner part of the cell and form a centrosome. Now, when it's near the nucleus, at some point it migrates to the ends of the cell, and I'm going to show you in just a second, and that aids in cell division. So I gave you some background on the tryptophan architecture. Now, these tryptophan networks exhibit a phenomena known as superradiance. This cooperative optical response to ultraviolet light is a light harvesting mechanism where tryptophan molecules, which are chromophores, interact in a coherent, which is synchronized, emission cascade, transferring, basically emitting absorbed photonic or light energy in the ultraviolet spectrum to neighboring tryptophan molecules, theoretically enhancing ultraviolet absorption by up to 4,000-fold. In simpler terms, sunlight is efficiently absorbed by us via amino acid highway of tryptophan molecules, and it's boosted and moved throughout the entire body to help facilitate optimum human biology. Now here's a diagram, or here's a video. And this experiment's using a vertical vibration plate of water, and you could see how as the vibration builds and builds and builds, water then is emitted. And it's the same concept, and they're trying to duplicate what superradiance is, but this is the only type of experimentation that they've come up with so far that could show it. So it's vibrating, building, and then ejecting that energy. So it's concentrating that energy to such a point where then it is emitted. Well, think about that. If it was a linkage of molecules in the microtubules, it's going to pass that concentrated energy along that chain. Pretty amazing stuff. And that architecture exists throughout our entire body. The superradiant property of tryptophan networks appears to be a conserved feature across evolutionary timescales. This suggests a fundamental role for tryptophan, its aromatic ring structure, and potentially other such molecules containing chromophores, melanin as an example, as fundamental molecular components for mediating cellular light interactions. Without this interaction, light and health fail. This is our direct connection to light and the natural world. And so the diagram below shows from 420 nanometers to 755 nanometers of light from the blue spectrum to the red spectrum. And then you could see the density from 1 to 3 millimeters of skin, and you can see that down by the 640 nanometer we're getting to the bottom of the hair follicle where melanin exists, so the light, that wavelength of light is being absorbed by melanin. Then you go a little bit deeper, there is red oxygenated blood, blue deoxygenated blood, a little fat, and so the red spectrum is absorbed deep within the system, and so hemoglobin, or blood vessels, as well as fat and other structures are absorbing light. And then you see to the far right, it says light beam. Some of it is being reflected off of the top layers of the skin. Then there's a little bit of scatter. The electrons are scattering around the area. And then the deeper it goes, we're getting actual absorption. Now, the diagram on the right, just to start with that, is, and I mentioned this before, in all of chemistry, there's only two reactions: oxidation and antioxidation or reduction. So you're either oxidizing something, which is losing electrons, or you're reducing something or antioxidizing it when you're gaining electrons. Lastly, these tryptophan meganetworks possess a photoprotective function, which suggests a mechanism where ultraviolet energy is dispersed throughout the protein network. This mechanism acts as a photoreducer, so an antioxidant in a way, adding electrons via photonic energy, or antioxidant for individual cells, nerves, and even the brain, preventing damage from concentrated light exposure as an oxidant. So it's kind of a self-regulating mechanism, so we can never really get too much. The body will disperse that energy. So again, I'll reread this last sentence. This mechanism acts as a photoreducer, add electrons via photonic energy, or antioxidant for individual cells, nerves, and even the brain, preventing damage from concentrated light exposure, which is an oxidant, or a generalized state of oxidative stress. This implies that cells under higher oxidative stress, a diseased state, might depend more on light and its interaction with tryptophan. Why all of this matters to you and me. Within this intricate protein architecture, a mega network of tryptophan molecules throughout the body could act as a quantum optical system. This system could facilitate the absorption, transfer, and emission of light and electromagnetic information. This process might optimize cellular function through increased electron levels, leading to a net negative charge and inherent antioxidant capability. This in turn could mitigate oxidative stress, potentially contributing to improved health and lifespan. Furthermore, this light-mediated process represents an ancient mechanism dating back to the endosymbiotic origin of eukaryotes. And eukaryotes are basically any cell with a cell membrane and nucleus, so plant and animal cells, potentially linking it to evolutionary cellular development and function, as well as maintaining an organism's health. This truly is a photophysical control of our biosystems. So into symbiosis, you could see moving from left to right, we had number 2 is a nucleus forming, and number 3 is the endosymbiotic event where an aerobic bacterium is absorbed within the cell engulfed, and that becomes the mitochondria, and then this would be then a plant cell, and the cyanobacterium would be engulfed, and that would be the chloroplast. So then this would be a plant cell. If it was minus the chloroplast, had it contained a nucleus, then it would be a animal cell.